Hello everyone, myself Shrishti Patil and here I am presenting a video about a research paper based on purification and biological evaluation of metabolites produced by Streptomyces species TKVL333. So, Streptomyces are a genus of bacteria known for their ability to produce a wide range of bioactive compounds. These compounds have a significant impact on various industries including agriculture, medicine, and biotechnology. Key characteristics of Streptomyces start with that they are soil-dwelling organisms, they have a complex life cycle, and that they are producers of secondary metabolites. Background of the research topic. Streptomyces species TKVL333 is a specific strain of actinobacteria. The researchers focused on isolating and characterizing the bioactive metabolites produced by this very particular strain. These metabolites hold the potential for various applications such as drug development or agricultural use. Streptomyces species are very well known for their ability to produce a diverse array of bioactive compounds. Therefore, this research was aimed to isolate and characterize the metabolites and to explore their potential as antimicrobial agents. The main methods used in this technique was starting with isolation being the first one. So, Actinobacterium strain TKVL333 was isolated on aspergillus glucose agar medium, incubated for 7 days at 37 degrees Celsius using a soil dilution technique. The strain was maintained on yeast extract, malt extract, dextrose agar medium, that is YMD, at 4 degrees Celsius for further study. Fermentation. Loopful culture of Streptomyces species was cultivated on YMD broth, also known as seed broth, and incubated on a rotary shaker at 30 degrees Celsius. After 48 hours of incubation, the results were obtained. Extraction. The first step involved in extracting the metabolites from the fermented broth was by using ethyl acetate. Guys, ethyl acetate is a common organic solvent which is effective in extracting various secondary metabolites from the microorganisms. The fermented broth likely contained the metabolites produced by the species. So, by adding ethyl acetate and mixing the solution, the metabolites partitioned into organic phase due to their higher solubility in organic solvents as compared to water. After extraction, ethyl acetate layer was likely separated from the aqueous broth layer which contained water soluble components. So the ethyl acetate extract then proceeded to the further stages of purification using chromatographic techniques. The culture filtrates obtained after cultivation of the strain in 2.5 liter conical flasks for 96 hours were extracted twice with ethyl acetate and concentrated to dryness under a vacuum at 35 degrees Celsius. The crude extract thus obtained was defatted with cyclohexane to give polar and non-polar residues. Purification. After extraction, the ethyl acetate layer was likely separated from the aqueous broth layer. So, then the further purification stages were done using chromatography techniques. So, there were three types of chromatography techniques used, starting from silica gel column chromatography, then thin layer chromatography, and the third one being high performance liquid chromatography. So, silica gel column chromatography is a common laboratory technique used to separate and purify mixtures of compounds. It utilizes a column packed with silica gel, a polar stationary phase, and a mobile phase, which has solvent or a solvent mixture. So the working is given, please do read. In context of the research paper, silica gel column chromatography was likely used to purify or further purify the extract obtained after the initial extraction with ethyl acetate. By passing the extract through a silica gel column with a suitable mobile phase, the researchers could separate the various metabolites present in the extract based on their polarities. Properties. Stationary phase was in which silica gel was used, which was a polar material. Mobile phase in which a solvent or solvent mixture was used. Sample loading, that was extract obtained after initial extraction with the ethyl acetate. Separation mechanism was the differential interaction of the metabolites with the silica gel based on polarity. Fractions collected, that is eluted components from the column at different times. Second is thin layer chromatography. Thin layer chromatography is TLC. So this is a simple and versatile technique used to separate and analyze compounds in a mixture. It's based on the principle of differential adsorption, where different compounds have varying affinities for the stationary and the mobile phase. The working is given, please to read. So in context with the research paper, TLC was likely used as a preliminary analytical tool for monitoring the purification process, assessing the purity of the isolated compounds, and optimizing chromatographic techniques. Third, high performance liquid chromatography, that is HPLC, also known as high pressure liquid chromatography, is a high resolution chromatographic technique that utilizes a column packed with a specific stationary phase and a mobile phase pumped at high pressure. HPLC offers excellent separation capabilities and can be used for analytical and preparative purposes. The working is given, please to read. In context with the research paper, HPLC was likely used as a final purification step to isolate individual metabolites from the complex mixture obtained after earlier purification steps like the silica gel column chromatography and the thin layer chromatography. So the key roles of HPLC in this research were about purification, identification, quantification, and by utilizing HPLC, researchers could achieve a higher degree of separation and obtain purer samples of the metabolites, enabling more accurate characterization and analysis of their properties. Bioassay guided purification. So this particular type of purification is used to isolate bioactive compounds from the complex mixtures. This involves combining the chromatography with biological assay to identify fractions containing the desired activity. So steps involved are extraction. Second is fractionation, which is the crude extract is subjected to chromatographic techniques like column chromatography to separate it into multiple fractions. Third is bioassay, that is each fraction is tested for its biological activity, that is either antibacterial, antimicrobial, anti-tumor, etc. Fraction pooling. Fractions exhibiting the desired activity are pulled together. Further purification, the pooled fractions undergo additional purification steps like HPLC to isolate the pure bioactive compound. Structure elucidation. The purified compound is characterized using spectroscopic techniques to determine its structure. So the relevance to the research paper of bioassay guided purification was that the testing of different fractions of crude extract for antibacterial activity against specific microorganisms was possible. Identifying fractions that exhibited significant inhibitory effects and subjected these active fractions to further purification steps to isolate the pure compounds and their specific bioactivities was also possible because of the bioassay guided purification. Result. The research successfully employed bioassay guided purification to isolate antibacterial metabolites from Streptomyces species TKVL333. Two compounds were isolated and purified using a combination of chromatographic techniques and bioassays. These compounds demonstrated antibacterial activity against Staphylococcus aureus and Escherichia coli. Conclusion. The study successfully isolated and characterized antibacterial metabolites from Streptomyces species TKVL333. The bioassay guided purification approach proved effective in identifying and purifying compounds with antimicrobial properties. The isolated compounds hold potential as lead compounds for the development of novel antibiotics. Further studies are required to elucidate the complete structure, activities, relationships, and evaluate the pharmacological properties of these compounds. References: this video was based on the research paper titled Purification and Biological Evaluation of the Metabolites Produced by Streptomyces species TKVL333. This research was done by the Department of Botany and Microbiology of Acharya Nagarjuna University, Guntur, and Organic Chemistry Division of Indian Institute of Chemical Technology, Hyderabad. Thank you.